Well, welcome everybody again, and thanks for joining us. We are very happy that you're here today with us, and it's going to be our 9th webinar in the GPT webinar series for 2021. It's September. I hope you're getting the red leaves around your household, wherever you are, and getting a cooler temperature as we go in through the year. And today is my great pleasure to introduce Dr. Steve Smith to deliver the webinar today. Steve is a new member to the GemPharmatech US team. Welcome, Steve, on board. And Steve did his undergrad study at Beloit College in Wisconsin. And then he went on to do graduate work at North Carolina State University in the Department of Microbiology, where he studied lipid biochemistry in Baker's Yeast in the lab of Dr. Leo Parks. And then for his postdoc research, he went on to Gladstone Institute of Cardiovascular Diseases in San Francisco, California, in the lab of Robert Fares, where he studied the genetic underpinning of cholesterol metabolism and triglyceride synthesis. From there, Steve started his industry career at Parke Davis Pharmaceuticals, where he continued to study diabetes and glucose metabolism. Steve joined GemPharmatech just a couple of months ago as our director of business development, where he will be help the company's exploration in the US market, serving our customers. So without further ado, welcome, Steve, and the stage is yours. For today's webinar, Steve is going to share with us the new insights that our R&D team has gained from the mouse models for pancreatic, breast, and colon cancers. Steve, welcome.
Thank you very much for that introduction, Zhiying. So thank you everyone for joining and attending today's webinar, and look forward to discussing these models with you. So today's webinar is actually a pre-recorded presentation. So beginning that. Hello, my name is Steve Smith. I'm the Director of Business Development at GemPharmatech. Thank you for joining us today to learn about GemPharmatech's spontaneous tumor models and that were developed for preclinical therapeutic research. Today, I will discuss several of these models and how they and their use may help accelerate your research studies. Cancer is an enormous global health problem and the second leading cause of death in the United States. It's estimated that 40% of the population will be diagnosed with cancer in their lifetimes. In 2018, there were 17 million new cancer cases and 9.5 million cancer deaths worldwide. By the year 2040, it is anticipated there will be 27.5 million new cancer cases and 16.3 million cancer deaths, simply due to the growth and the aging of the world population. Thanks to all the research programs, approximately 2/3 of cancer patients survive at least 5 years. In addition to the therapeutics, there have been improved diagnostic tests and improved lifestyle changes. However, many patients die of cancer regardless, and many cancers cannot be treated. Research scientists need to continue discovering drug targets and developing new therapies. Nearly half of new cancer cases are caused by prostate, lung, and colon cancers in men, and breast, lung, and colon cancers in women. Nearly half of all cancer deaths are due to lung, prostate, colon, and pancreatic cancers in men, and lung, breast, colon, and pancreatic cancers in women. Sadly, the cancer epidemiology has not changed significantly in the past 50 years. Preclinical discovery and development of novel oncology therapeutics and targets has utilized a range of murine models to attain the therapeutic successes currently used in the clinic. These models include xenograft tumor models, where immortalized and patient-derived human tumor lines are implanted in immune-competent mouse strains, genetically engineered mouse models with humanized immune systems to model the human immune response to the tumors as well as the immune response to therapeutics. And spontaneous tumor models, where tumors spontaneously appear and grow due to the same mutations driving human disease. These are in immune competent mouse models. There is great research utility in using spontaneous tumor models to help discover and develop new therapeutics as well as targets using immune competent mouse models. Today, I'll be discussing some spontaneous tumor models, including pancreatic, breast, and colon cancer models. The epidemiology of pancreatic cancer is exceedingly troubling, in a large part due to the disease not being diagnosed until relatively late stages. When diagnosed early and the disease is localized, 35 to 40% of patients survive for at least 5 years. However, pancreatic cancer is most frequently caused by mutations in P53 and Kras genes, and unfortunately, it is found very late and it tends to be disseminated throughout the pancreas. This slide shows the current treatments for the pancreatic cancers, which are being tested in the clinic. The highlighted treatment strategies are examples of small molecule chemotherapy combined with immune checkpoint inhibitors to treat pancreatic cancer. These are currently in very early stages, phase 1 and phase 2. GemPharmatech has generated 3 models of pancreatic cancer to faithfully represent human disease. As mentioned, pancreatic tumors are found to result from mutations in Kras, P53, and often both mutations in many patients. GemPharmatech's KPC model is in the C57BL6 background, and it's a very powerful oncology model for carrying the inducible mutations in both Kras and P53, which these mutant genes are then expressed when the KPC strain is crossed with the PDX1 Kras strain, with all animals, all offspring then developing pancreatic tumors, typically by 8 to 16 weeks of age. The images below show the massive pancreatic tumors in the KPC strain. Both the Kras G12D and P53 R172H models develop pancreatic tumors after being crossed with the PDX1 Kras strain. This model expresses Kras behind the pancreatic duodenal homeobox 1 promoter. When crossed with the Kras or P53 models, the Kras causes the upstream LOXP stop LOXP sites to be excised and the mutant alleles to be expressed, causing pancreatic tumorigenesis. On this slide, you can see the histology of the tumors in the KPC model and details of the progression. These tumors, again, begin to first be observed in animals at 8 weeks of age and continue out beyond 16 weeks of age, with animals surviving beyond 16, sometimes to 20 weeks of age, but with very increasing dramatic tumor burden. Since tumor development and metastases in the KPC model is quite aggressive, and like human disease, it is difficult to know the disease stage, this model has found excellent utility in serving as a donor for syngeneic tumors. The tumor growth, metastatic development, and tumor histology is consistent from the original tumors here in PA, the upper figures, out beyond 5 passages of the tumor cells grown in culture, and then cycled as well through additional animals. And you can see that after 5 passages, that the tumor growth is still very consistent. These are individual animals here in the tumor growth chart. Individual animals display the subcutaneous tumors. This publication from 2018 deals chemotherapeutics that have been tested in pancreatic ductal adenocarcinomas in preclinical mouse models. This publication specifically highlighted immuno-oncology therapeutics tested in combination with other immuno-oncology therapeutics, standard of care small molecule therapeutics, and radiation therapy. In studies monitoring tumor growth and response and changes, as well as the changes then in body weight due to therapeutic intervention in sub-Q implanted syngeneic KPC tumors, these animals had 6 animals per group. And you can see that the best tumor growth suppression and response was observed in those animals receiving gemcitabine at 100 mg per kg, shown in red, as well as gemcitabine plus anti-OX40, which is shown in purple, and gemcitabine plus anti-OX40 antibodies plus anti-PD-1 antibodies in brown. As you can see from the excised tumors at the end of the study on the right, that the gemcitabine alone tumors in G2, as well as the combination therapies in G6 and 7, the tumors themselves are notably much smaller, consistent in size and much smaller than those receiving other therapeutics. This slide shows the KPC tumor line development. As anticipated, a larger inoculum results in a more rapid and larger tumor growth, as shown on the left. Syngeneic tumor cells can be implanted IV for modeling tumor metastases, or sub-Q for monitoring tumor growth alone for solid tumors. Using an immune competent strain expressing the human PD-1 and or PD-L1 can be very valuable for exploring immuno-oncology therapeutic treatments in this model. The Kras mutations are found at high frequency in human cancers, including pancreatic, colorectal, non-small cell lung cancers. Our Kras G12D is an authentic model of human cancers following the crossing with a Cre-expressing, Cre-recombinase expressing line. Typically, tumor model mice survive beyond 16 weeks of age, but do display increasing and dramatic tumor burden by that point. Shown in the top row are lung tissue samples from standard Kras G12D that were not crossed with the Cre line. So there are no tumors that appeared or were visible and found in these animals. The middle line of histology sections shows spontaneous tumors in mice resulting from crossing the Kras G12D with the LYZ2 Cre driver strain, resulting in tumors in myeloid cells, shown here with metastases in the lungs. LYZ2 encodes the lysozyme 2 gene. When the Kras G12D strain is crossed with the LYZ2 Cre strain, tumors are observed in lungs, spleen, and other organs. At the bottom are images from the Kras G12D that were crossed with the ALB Cre strain, resulting in spontaneous tumors in the liver. The albumin promoter drives liver-specific expression of Cre recombinase. This model, or these models, are very good models for studying in vivo pancreatic, liver, small cell lung cancers. And you can screen the efficacy of small molecule anti-tumor therapeutics with this model. The tumor suppressor gene, P53, regulates transcription of numerous genes, which are involved in cell cycle arrest, apoptosis, DNA repair, senescence, and metabolism. Over 50% of all cancers are found to have loss of function mutations in the P53 gene, resulting in this being the primary driver of disease, or also a contributing mutation, making the tumors more difficult to treat and more aggressive. Our B6 P53 knockout model, which is generated in the C57BL6 background, has great utility for modeling human cancers in many organs. By 3 to 6 months of age, mice develop tumors in many tissues, including malignant mesenchymal fibrosarcomas, as well as mesenchymal epithelial sarcomas in soft tissues. GemPharmatech also has a P53 conditional gene knockout strain, which can be crossed with Cre driver lines, resulting in cancers arising in select tissues for your research purposes. This B6 P53 knockout strain can be used for screening chemotherapeutics for combating disease and looking at following the efficacy of your small molecule and antibody therapeutics. GemPharmatech has developed several breast cancer spontaneous tumor models driven by mutations frequently responsible for human disease. In an effort to more effectively combat breast cancer, multiple immunotherapy approaches have been exploited in very recent past. And these are primarily through immune checkpoint inhibitors, including the PD-1 and PD-L1 antagonists. These checkpoint inhibitors are used to treat patients depending on the breast cancer subtype and mutations identified that are driving the human tumor growth. While some antibody therapeutics may be effective on their own in certain cases, in certain cancers, antibody therapeutics combined with standard of care therapeutics are being employed with the hope of greater efficacy and reduced toxicity. There are several genetically engineered breast cancer models that have been found to have great utility in preclinical research studies. In addition to the Kras and P53 mutant strains, there are additional spontaneous tumor models that are excellent for modeling human disease. The MMTV-PyMT breast cancer model uses the mouse mammary tumor virus promoter to drive the polyoma virus middle T antigen. Elevated expression of the PyMT results in downstream expression of the ERB2 and Wnt1 oncogenes in their pathways, resulting in mammary tumors closely resembling the human breast cancer in many patients. Shown in these images are orthotopically implanted syngeneic tumors and their consistent growth following implantation in the animals. The MMTV-PyMT is an aggressive tumor model with primary tumors being identified as early as 6 weeks of age in both male and female mice, and metastatic tumors identified in many tissues several weeks later. The MMTV-PyMT strain is an excellent model of primary breast cancer as well as metastatic tumors. Next, I'll be discussing the colon cancer spontaneous tumor models that have been developed by GemPharmatech. As mentioned earlier, colorectal cancers is the cause of nearly 10% of deaths and diagnoses in men and women worldwide. Diagnostic procedures and therapeutic options have improved the prognosis of human colon cancer and have improved along with the improved diets and healthier lifestyles, have improved outcomes. However, there's still a very large number of patients who, for whom the therapeutics do not respond. There are 5 stages of colon cancer growth, and treatment success depends on the tumor stage when it is discovered. In stage 0, the tumors are very early in diagnostics. They have not yet grown beyond the inner mucosal layers of the intestine. In stage 1, the tumors have grown through the mucosa, but not yet spread into the lymph nodes. Several substages in stage 2 characterize the tumors crossing the outermost layers of the wall of the colon or rectum and growing through the wall itself. Cancer is not yet spread to the lymph nodes in stage 2. In stage 3, also having several substages, and these are characterized with a greater tumor growth and greater spread into the surrounding lymph nodes. In stage 4, which also has several stages, and is determined by the extent of the metastases throughout the body and extent throughout and spread throughout the lymph nodes. All colon tumors are caused by and driven by genetic mutations. Often the mutation is in the APC gene, which can result in a cascade of additional mutations, destabilizing cell division and resulting in uncontrolled cell growth. Development and metastatic spread of the colon cancers here is showing the collection of genetic mutations as the tumors themselves grow. The APC, or adenomatosis polyposis coli gene, encodes a tumor suppressor protein that acts as an antagonist of the Wnt signaling pathway, controlling Wnt signaling. Altered expression of the APC and then downstream beta-catenin and TCF mediate Wnt pathway dysregulation and are the cause of many human colon cancers. Reduced expression of the APC protein can help lead to beta-catenin not being degraded and free beta-catenin accumulating in the cytoplasm and in the nucleus, leading to carcinogenesis. APC proteins are also involved in cell migration, adhesion, transcription, and apoptosis. Oops. The current immunotherapy standards of care for colon cancer are Opdivo or nivolumab, which is produced by Bristol Myers Squibb, and it's an immune checkpoint inhibitor blocking PD-L1. Keytruda or pembrolizumab is from Merck, and it binds and blocks PD-1. And in a combination with Opdivo and Yervoy, which is ipilimumab from Bristol Myers Squibb, which is a CTLA-4 inhibitor, combination allows immune checkpoint systems to attack tumors. However, despite these being standard of care and making it into the clinic and finding some utility, the current median progression-free survival rate for patients is barely 1 year in the best, and that's only for about half the patients. The APC MIN model was developed by GemPharmatech, and it results in spontaneous colon cancer that is a good representative of human disease. This model has a mutation in the APC tumor suppressor gene, and adenomas are found after 8 weeks of age, and nearly all animals display dramatic tumors by 15 weeks of age. As cancer develops, mice gradually develop cachexia, chronic inflammation, and anemia. The APC MIN model can be used for screening the efficacy of therapeutics and other drugs for the treatment of colorectal cancer. Shown here is a list of our 12 spontaneous tumor models. 11 of these models are currently available, and 1 is still in the final stages of development. GemPharmatech has also developed over 200 Cre recombinase expressing mouse lines that can be used in combination with 7 of our spontaneous tumor models to induce spontaneous tumors in tissues of interest. GemPharmatech can also develop custom mouse models for your research purposes. Thank you for joining us today to learn about the spontaneous tumor models developed by GemPharmatech. Please contact us with any questions you may have regarding these mouse models, our immune-compromised mouse models, and our humanized models, as well as custom mouse model development. Thank you.
All right. I think Steve is coming back to join us live again, and I'm checking in the Q&A and the chat box to see whether we have got questions. I see Steve has unmuted himself.
There we go.
Great. Glad you're back. So
yeah, I made it.
exactly in time. Right. So I do see there's a question regarding the appearance of tumor among the animals. This is about the B6 KPC, I believe. The question is, do tumors appear in all animals at the same age, for example, 8 to 10 weeks of age?
It's a very high percentage by 8 to 10 weeks, and by 14 to 16 weeks, there are tumors found on all animals. Spontaneous tumors arising in all animals, and the tumor burden can get very considerable after 16 weeks of age, and often due to IARC rules and guidelines, animals need to be taken down by that point. And so, but they do arise and metastasize very similar to human disease in the models.
I see, but I guess in humans, the progression will be much longer, right? From the appearance to the metastasis.
It would, yes, indeed. And so certainly within humans, one of the confounding problems with cancer is that it's not detected until it's too late, or until it requires considerable surgical intervention, and then hoping that the therapeutic approach can actually access and treat all of the primary tumors as well as any metastases that have formed.
I see. All right. So, the same person asked, saying, so in this case, can you still keep the colony as a standalone breeding colony to maintain the animal?
With most of the models, you can, but it can be difficult. I believe the models are all maintained. Most all of them are maintained as heterozygotes, heterozygous models, and it does take rapid turnover of the breeders, because breeders will become cancerous and be unable to continue breeding, or may have to be taken down after a couple litters.
I see. Great. Well, thanks for the information. Another question I see is about, do you have other breast cancer models in your development or in your pipeline?
Currently, we do not. We're just the MMTV model is our, of the ones that were mentioned there. However, with the P53 as well as Kras models, if they're the ones that are the LOX-P controlled, if they're crossed with the proper Cree driver line, you can use those for a breast cancer model as well. In fact, we have I believe it's a whey acidic protein promoter driving Cree recombinase, and that, if that's crossed with the P53 or the Kras, that will result in breast tumors as well.
Great. Well, thanks for the information, Steve. I see one more question, and I'm going to read it out, but I think this could be a tough question. Ooh. So how well does the disease pathology in mice recapitulate human diseases?
That is a toughie. And like a lot of preclinical models, translational research models, it depends. And it depends on what you're looking at and what your goals are. Certainly with these models, having the primary driver mutation, like a Kras or P53, for instance, driving tumor development is very, very similar to the human disease. And additional mutations that accumulate to drive the disease are very, very frequently in these spontaneous models, is also quite similar to the human condition. And so then it's a matter of, because this is these are immune competent animals, you can utilize immune checkpoint inhibitors, again, needing to be certain that they're going to bind the mouse proteins for activity, but they can at least give a very good indication if you are screening drug targets. It allows doing some efficacy studies as an animal model
with these.
I see. Well, since you mentioned the immune checkpoint inhibitors, I do want to just mention, as I have been sitting in quite a few of our webinars, GemPharmatech does have a series of humanized immune checkpoint mice for research, especially preclinical screening purposes. So I've been checking, and I don't see any other questions coming in. So thank you again, Steve. And before I close today's webinar, I just want to share a couple of provide this information to everybody who come today to join us. We are running a new promotion for our flagship pipeline, which is called the Knockout All project. If you're familiar with our products and services, you may know this already, KOAP. If you haven't heard about it today, we are providing you the information. So this project is a project aiming to knock out or conditionally knock out every protein coding gene in the mouse genome for the purpose of research, understanding the gene function, starting from the mouse, and then translating it to human health and disease. For this particular promotion, well, we have already developed 17,000 KO and CKO by now. So there's a big pool for you to select from. The offer today, well, not just today, starting from today, you can choose from two options. One is if you buy 5 strains out of the KOAP project, you pay the price of 4. Or if you don't have the capacity to get this many all at the same time, you can use our referral program where if you have you become our customer in the past, you can refer your friends, and both of you will get a discount, which can be up to 20% if you are new to us. And I believe you can still take advantage of the referral program by partnering with our existing customers. And then second piece of information I want to bring to your attention is that as GemPharmatech US grows, we have had live colonies in the States. And in particular, I can let you know is in Boston. If you are in the Boston hub, it's very close to you. In particular, out of these 20-some strains, we have 17 of them are humanized immune checkpoint models. The popular ones, I can tell you right away, the PD-1, PD-L1, CTLA-4, LAG-3, TIM-3, and a few others. We also have the B6, the BOB-C backgrounds, as well as double and triple targets. I can mention another one, 41BB, CD73, for your selection. And second thing, I do want to give a highlight for the immune or the severe immunodeficient models. Our NCG has been serving customers in the States and in Europe for more than 6 years up to this point. We have our partner CRL being our distributor or our dedicated distributor, helping us providing this NCG mouse to the customers in North America and Europe. And we do have a few others, which I want to highlight. We are putting an effort to the Alzheimer's disease research. We have a new model under development. We also have 5 different humanized ACE2 mouse in our portfolio as a toolkit for people who are engaged in the COVID-19 research. We believe humanized ACE2 mice are a great tool to help the pandemic research. We have them on the B6, on the BOB, as well as on the NCG background. So just information to you. So check us out on the website, GemPharmatech.us, and also at any time you can email us at sales@GemPharmatech.us, and our team member will reach out to you as soon as possible. And again, well, thanks again, Steve, for the webinar, and thanks everybody who joined us today at today's webinar. So before we close, any last words from you, Steve?
No, I think covered just about everything with these models. I see there was one last question that came in about the availability of this webinar. And yes, it will be available online, and we will just log on online with this or connect with us online for this presentation, and we will send you a link to the presentation. Again, both Zhiying and I, as well as the rest of the technical team here at GemPharmatech, would be very happy to speak with you about any of the models discussed, or custom model development, or any other models that we have that have already been developed.
Well, actually, I have to say thanks for the question. This reminds me, our October webinar is coming along, is in preparation. The date will be October 26th. The topic will be NASH, animal model or mouse models for NASH research. And stay tuned, check out our website. We will be posting on the website as well as our LinkedIn accounts. We do have Instagram, Facebook, and Twitter. If you want to hear from us, you have all the tools that you can use. And thank again, and have a good day.
Great, thank you.
Bye-bye.
Bye.